Acne is not just a teenage problem. Research shows that adult acne now affects up to 50% of women and 25% of men in their 30s, and the rates are rising. Separately, skin laxity and early wrinkle formation are concerns appearing in people's late 20s and early 30s. What most people do not realize is that both conditions, acne and premature skin aging, share underlying biological drivers. And some of the most powerful interventions for both are the same: dietary changes, targeted supplementation, and specific metabolic habits like intermittent fasting. A randomized controlled trial published in the American Journal of Clinical Nutrition found that young men who adopted a low-glycemic diet for 12 weeks showed significantly fewer acne lesions than controls eating a high-glycemic diet, with acne scores dropping by nearly 24%. The mechanism was lower insulin and IGF-1, hormones that directly stimulate sebum production and follicular keratinization, the two processes underlying acne formation. This study established that acne is a metabolic disease as much as a skin disease, and that diet is a primary driver, not just an aggravating factor. This video draws on dermatology and nutrition research. Smith and colleagues' 2007 ACNE trial established the dietary acne link in a rigorous RCT. Burris and colleagues reviewed the medical nutrition evidence for acne comprehensively. Research in oxidative medicine and cellular longevity documented the role of autophagy, activated by caloric restriction and fasting, in skin cell health and aging. And Proksch and colleagues' 2014 double-blind trial confirmed oral collagen peptide supplementation for skin elasticity. In this video, we are going to see the complete research-supported approach for both acne-free and firm, younger-looking skin. The dominant model of acne treatment is topical: apply a cream, take an antibiotic, or use a retinoid. These are valid interventions. But the research increasingly shows that they treat symptoms without addressing the underlying metabolic drivers. Similarly, the conventional skin aging model is primarily cosmetic: apply products to the surface. But research shows skin health is fundamentally systemic, determined by blood glucose management, hormonal balance, nutritional status, and metabolic processes that topical products cannot reach. Both acne and premature aging require an inside-out approach supported by the evidence. The mechanism connecting diet to acne is now well characterized. Refined carbohydrates and high glycemic foods spike blood glucose, which triggers an insulin surge. Elevated insulin and its downstream signal IGF-1 activate androgen receptors in sebaceous glands, increasing sebum production. Excess sebum combined with follicular hyperkeratinization, also stimulated by IGF-1, creates the ideal environment for Cutibacterium acnes proliferation and inflammatory acne. Research has confirmed each step of this pathway. Dairy, particularly skim milk, independently raises IGF-1 and has been associated with acne in multiple studies. Research comparing two dietary patterns on skin health reveals dramatically different biological outcomes. The high glycemic, dairy-heavy, processed food pattern produces chronically elevated insulin and IGF-1, which stimulate acne, while simultaneously promoting advanced glycation end products, sugar collagen crosslinks, that accelerate skin aging. The low glycemic, whole food, anti-inflammatory pattern keeps insulin and IGF-1 at lower levels, reducing sebum stimulation, while providing antioxidants that prevent glycation and collagen oxidation. These are not two sides of an opinion debate. They are two measurable hormonal and molecular environments with documented skin consequences. The Smith 2007 ACNE trial is the best-designed dietary intervention study for acne. 43 males aged 15 to 25 were randomized to either a low-glycemic low diet or a high-glycemic control diet for 12 weeks. Acne lesion count fell significantly more in the low-glycemic group, by 23.5% versus 12% in the control group. Simultaneously, IGF-1 levels were significantly lower, and insulin sensitivity improved in the low-glycemic group. The researchers also noted decreased androgen activity, lower free testosterone and DHEAS, in the intervention group. Diet had changed the hormonal environment driving acne. Intermittent fasting connects to skin health through autophagy, the cellular self-cleaning process. During fasting periods of 12 to 16 hours or more, autophagy in skin cells increases significantly. This process degrades damaged proteins and cellular debris, including the dysfunctional organelles that accumulate with age and impair skin cell function. Research in dermatology has shown that autophagy is specifically impaired in photoage skin. Restoring autophagy through fasting and caloric modulation may therefore provide a cellular rejuvenation signal that skin cells are designed to receive, and that constant eating chronically prevents. Research on wrinkle prevention and treatment has identified specific interventions with documented effects on skin structure. Oral collagen peptides, 2.5 to 5 grams daily, show significant elasticity improvement in double-blind trials. Topical retinoids increase collagen gene expression in fibroblasts and reduce collagen-degrading enzyme activity. Vitamin C supplementation above adequate levels, typically 250 to 500 milligrams daily, ensures maximum collagen synthesis cofactor availability. And daily sunscreen use is the single most evidence-backed wrinkle prevention measure available, preventing the UV-driven matrix metalloproteinase activation that destroys collagen daily. Zinc is relevant for both acne and skin aging through different mechanisms. For acne, zinc has direct anti-inflammatory and antibacterial properties against Cutibacterium acnes, the primary acne-causative bacteria. Multiple clinical trials have tested oral zinc for acne with positive results, though typically not as strong as prescription options. For skin aging, zinc is a cofactor for collagen synthesis and for enzymes involved in skin repair. Research on zinc glycinate and zinc gluconate supplementation shows anti-inflammatory effects in acne-prone skin, particularly in deficient individuals. Zinc adequacy is a prerequisite for optimal skin biology. The research framework for both clear and firm skin operates on 3 simultaneous levels. The internal level addresses diet and metabolic health, eliminating high glycemic foods and excessive dairy to reduce insulin and IGF-1, the primary acne drivers. The cellular level uses intermittent fasting to activate autophagy, the skin cell renewal process suppressed by constant eating. And the structural level uses specific supplements and topical interventions, collagen peptides, vitamin C, zinc, and retinoids, to provide the building materials and signals for collagen synthesis and skin repair. These 3 levels are complementary and work best together. Research documents different timelines for acne and skin firming interventions. For acne driven by diet, low glycemic eating shows measurable lesion reduction within 4 to 6 weeks, reflecting the hormonal changes occurring as insulin and IGF-1 normalize. Zinc supplementation shows acne improvement at 6 to 8 weeks in clinical trials. For skin firmness, collagen peptides show elasticity improvements at 8 weeks in double-blind trials. Retinoid use shows structural dermal changes, measurable on imaging, at 3 to 6 months. The fastest-acting interventions for both conditions are the dietary and metabolic changes that address the hormonal root. Based on the research, here are the specific steps for clear and firm skin. Eliminate refined sugar, white bread, white rice, and high glycemic processed foods. This is the foundation of both acne control and anti-aging. Reduce or eliminate dairy, particularly skim milk. Practice a 14 to 16-hour daily fasting window to activate skin cell autophagy. Take 2.5 to 5 grams of hydrolyzed collagen peptides daily. Ensure adequate vitamin C intake, 250 to 500 milligrams daily. Introduce a retinol product at night gradually. Apply SPF 30 or higher every morning. This single step prevents the UV collagen destruction that undermines all other efforts. The research logic for both acne and aging is rooted in the same metabolic biology. If your diet is high in refined carbohydrates, then insulin and IGF-1 stay elevated, simultaneously triggering sebum overproduction for acne and glycation of your collagen for wrinkles. If you shift to a low glycemic diet, then both processes slow immediately. If you add intermittent fasting, then skin cell autophagy activates, clearing the cellular damage that drives both acne bacteria proliferation and skin aging. If you add collagen peptides and retinol, then the structural rebuilding that diet and fasting enable has the raw materials to proceed. Acne and premature skin aging share deeper roots than most people realize. Both are driven by chronic insulin and IGF-1 elevation from high glycemic diets. Both are worsened by suppressed autophagy from constant eating. And both respond to the same core interventions: low glycemic eating, intermittent fasting, antioxidant nutrition, and targeted supplementation. The research has established the mechanisms clearly. Your skin is not a cosmetic concern. It is a metabolic organ that reflects your internal biology. Change the biology, and the skin changes with it. Your skin is a metabolic organ, and it responds to the same inputs that your heart, brain, and liver respond to. Start with the dietary foundation. Remove refined carbohydrates and high glycemic foods for 4 weeks and observe your skin. Add a daily fasting window. Consider collagen peptides in the morning. Begin retinol at night. Protect your skin from UV every morning. These are not separate beauty tips. They are a coherent, research-supported approach to both clear and firm skin from the inside out. Your skin will tell you whether it is working. The research suggests it will.